start of Lecture 2, we'll begin by talking about mutations. A mutation is defined as any change in the DNA sequence of a chromosome. And these mutations come in three different types. The first is a substitution, or a point mutation. This is simply substituting one nucleotide at a base pair for another nucleotide, for example, an adenine for a guanine, or a cytosine for a thymidine nucleotide. This is the most common type of mutation, and it's important to realize that the total number of nucleotides in a given gene will not change with this type of mutation. The other two types of mutation actually change the numbers of base pairs in a gene. The first is an insertion mutation. This is a mutation in which one or more base pairs are inserted into the sequence of a gene. The second is a deletion mutation. This mutation is where one or more nucleotides are deleted or removed from the sequence of a gene. Now, substitutions also come in different types. Substitutions can either be transitions or transversions. A transition is the replacement of a purine nucleotide, which are your adenines and guanines, by another purine nucleotide, or the substitution of a pyrimidine nucleotide, which is thymidine or cytosine, by another pyrimidine nucleotide. Transversions are the opposite of that. Transversion is the replacement of a purine nucleotide, an A or a G, by a pyrimidine, a T or a C, or vice versa, and that's illustrated schematically at the bottom of this slide. There are three potential consequences of any nucleotide substitution. The first is silent mutations. These are mutations that don't change the coded amino acid in the gene. And these are most commonly found at the third position of the codon. If you look at the codon table, in which you have the nucleotides outlined for each of the amino acids in a gene or in a protein, you'll see that many of the amino acids are coded by multiple codons, and many of these have the third position in common. And so mutations at the third position of a codon often don't change the amino acid that's being coded by that particular codon. An example is shown in the slide here. In the first mutation on the left, you see that there's a uracil. This would be RNA, of course. A uracil is substituted by a cysteine nucleotide. In this case, it does not change. The codon still codes for cysteine just like it did before. Similarly, in the mutation on the right, you see a cytosine in the third position of a glycine codon being changed to a guanine nucleotide, which is also in the third position of the same codon. And that still encodes for the glycine amino acid, and so there's no change in the amino acid sequence there. Now, another type of substitution mutation is what we call a missense mutation. This is a mutation in which the encoded amino acid is changed. That is, that the codon changes from a codon encoding one amino acid to a codon which encodes a different amino acid. These most commonly happen at the first and second positions in the codon, although they can occur in the third position as well. Now, you can see that these can be either conservative changes or they can be non-conservative changes. For example, in the mutation on the left, we change a cytosine nucleotide at the first position of this leucine codon to a guanine, and that changes the codon from one that codes leucine to one that codes valine. Now, because leucine and valine are both hydrophobic amino acids, this is unlikely to change the structure or the function of this protein significantly, and so we call it a conservative mutation. On the other hand, the mutation that you see on the right, which converts a guanine nucleotide in this arginine codon to a uracil nucleotide, in this case changing arginine encoding codon to an isoleucine encoding codon, you can see that that would change the function of that codon. You go from a positively charged arginine amino acid to now a hydrophobic and neutral amino acid isoleucine. This is much more likely to change the structure and or function of the protein in which this amino acid resides. And so these we call non-conservative changes because they change one type of amino acid to a completely different type of amino acid. The third type of substitution mutation that we can see is what we call a nonsense mutation. This is a mutation that changes a regular codon to a stop codon. The consequence of this is that you get a truncated protein. For example, in the sequence that we show below, you have a uracil codon in the third position of a cysteine codon being changed to an adenine. And this changes the cysteine codon now to a stop codon. So this protein then, when it's being translated, would be truncated prematurely, and you would end up with a smaller protein than you normally would have. And this usually has functional consequences because it completely changes the structure of that protein. Now, insertion and deletion mutations are much more likely to have deleterious effects on the protein that's being encoded. That's because insertions or deletions generally cause frame shifts. What happens in this is you remove one or more nucleotides, and that shifts the codon frame such that now everything downstream of that mutation is now encoding a different amino acid than it originally did. We show an example at the bottom of this slide. Here you have a 6-codon nucleic acid sequence. In this, you'll notice that in the third position of the second codon, we have inserted an adenine nucleotide. So now instead of reading methionine, histidine, glycine, cysteine, glycine, arginine, proline, you can see that everything downstream of that mutation has been changed. Now it reads methionine, glutamine, tryptophan, valine, tryptophan, glutamine, threonine. So you can see that everything downstream of that would be different, and the function and the structure of that protein would be completely different. Deletions can also cause frame shifts. In the example that you see below, the second nucleotide of the histidine codon, which is an adenine, is removed from the sequence. And that changes it from encoding methionine, histidine, cysteine, glycine, arginine, proline to now encoding methionine, leucine, valine, alanine, and aspartate. And you can see again that the amino acid sequence has changed, and the structure and function of that protein has probably completely changed as well. The last thing that an insertion or deletion mutation can do is cause the formation of a nonsense codon. For example, in the sequence that we've shown below, we've inserted an adenine codon between the second and third nucleotides of that histidine codon. So now that we have that adenine nucleotide there, rather than reading methionine, histidine, cysteine, aspartate, arginine, proline, and continuing down the sequence, now it reads methionine, glutamine, leucine, and then a stop codon. This results in the formation of a truncated protein, similar to what we see in nonsense substitution mutations. Now, what you should notice about all of these is that something different would occur if the insertion or deletion comes in multiples of 3 nucleotides. So, for example, if we inserted 3 nucleotides or we deleted 3 nucleotides, the frame of the sequence would not change. That is, the codon structure wouldn't change. We'd just be removing one amino acid encoding codon. And so the sequence wouldn't change. You would just have one fewer amino acids in the protein that's being encoded. So let's do a little quiz on mutations. I have 2 sequences on this slide. Sequence 1 represents the wild-type mRNA sequence that's encoded by some gene. Sequence 2 is the same sequence, except it carries a single mutation. Now you can compare those 2 sequences and see where the mutation is. And then I'd like for you to consider how you would characterize this mutation. Would it be A, silent mutation, B, a missense mutation, C, an insertion mutation, D, a nonsense mutation, or E, a deletion mutation?